Hello students, hope you are doing good. Now here I'm going to discuss the second part of the chapter 2, sexual reproduction in flowering plants. So previously in the first lecture of this chapter, if you remember, we have discussed about anthers, okay, and stamens, the male reproductive part of a plant we have discussed. So in this slide, firstly we are going to in the coming slide, we are going to discuss about the female reproductive part. So let's get started. So the female reproductive part is referred to as gynoecium. So what gynoecium consists of? It consists of pistil. Okay. So again, the female reproductive part is gynoecium, and the gynoecium consists of pistil. Okay, so just a recap of the diagram of a flower. You can see over here, this yellow portion, this whole yellow portion is pistil. So this surface is stigma, this long filament is style, the round structure is ovary, and inside it is ovule. So together it is called as a pistil. Okay. Now pistils, there can be one pistil or there can be two or more than two in a plant. So a single pistil is referred to as pistil only, and if multiple pistils are there, that is referred to as carpel. So you can say collection of pistils is referred to as a carpel. So on the basis of like the presence of single pistil or multiple pistils, the plant can be categorized as monocarpellary and multicarpellary. So mono means single, carpellary means the carpel. So means it is having single pistil. Multicarpellary means it is going to have more than one pistil. Okay. Now coming to the next thing, this multicarpellary pistil means containing more than one pistil. It can be of two types. It can be syncarpous or apocarpous. Syncarpous means that the pistils are fused. Apocarpous means pistils are free. Now what do I mean by saying that pistils are fused? You can see over here, see, these are the pistils, these are fused in case of this plant, which is papaver. Here you can see the flower. Can you see this portion? All right, so this is enlarged over here. The pistils are fused with each other. All right. And in another example, you can see the pistils are free, like in Michelia. See over here, these pistils are free like this. This portion you can clearly see. This is Michelia. And the pistils over here are not attached to each other. They are freely existing. All right. Now coming to the next structure, which is ovary. So ovary has a cavity which is called as ovarian cavity, and it is referred to as a locule. Now placenta is located inside the locule. Placenta, you know, it is a tissue which provides the nutrition to the developing embryo. So the placenta is inside this cavity which is called as locule. And the ovules or megasporangia, one and the same thing. The another name of ovule is megasporangia. It arises from this placenta only. So you can see this diagram to clarify this. See, this is the ovule, okay, and this is the ovary wall. So this ovule is attached to the ovary wall with the help of placenta, and the cavity which is present inside the ovary is referred to as locule. Now the number of ovules which are there, they vary from one plant to the other. Like single ovule is present in wheat, paddy and mango, and many ovules, more than one, are present in papaya, watermelon and orchids. So you can see diversity. So now let us understand the structure of ovule very, very carefully. So ovule is referred to as megasporangium. Okay. Now ovule, it is a very small structure and it is attached to the placenta by a stalk which is called funicle. Okay. So ovule is there and placenta is there. Suppose this is ovule and this is placenta. it is attached to the placenta with the help of funicle. Okay, so this is not the just a schematic representation I'm showing you. Okay, the ovule, it is attached to placenta by a stalk which is called funicle. Clear? Now ovule is going to fuse with the funicle. It is going to fuse with this funicle, and the region where it fuses is called as hilum. So hilum is the junction between ovule and funicle. So coming to it again, if suppose this is ovule, this is placenta, so this is funicle which is joining the two. Now if this ovule fuses with funicle, then in the area, this region where it is going to fuse, this region is called as hilum. And there are protective layers of ovule also. So those protective layers are called as integuments. And integuments, they encircle the whole ovule except for a small opening which is micropyle. So suppose this is the ovule and this is the protective layer. Okay, so it is this protective layer is called as integument. Now except for a certain this area where small opening is there, the integument is present all over except this small opening, and this small opening is referred to as micropyle. The end which is opposite to this micropyle, this end, is called as chalaza or the basal part of ovule. Okay, so these terms should be very much clear to you. In the diagram, it will be more clear. See over here. Suppose if this is the ovule, the structure of ovule, okay. So this is the funicle which is helping the ovule to attach to placenta, and here the ovule is getting fused with funicle. Okay, this region is called as hilum. Can you see integument? Inner integument, outer integument means protective layers are there. There is one end where protective layer is absent, this opening. This is called as micropyle, and the end which is opposite to micropyle is the chalice. Okay. Then can you see a collection of cells which is nucleus? Yes, it is the source of abundant reserve food material, and embryo sac is located inside this nucleus. So this is embryo sac, and here collection of cells are there which is nucleus. So inside the nucleus, embryo sac is present, or it can be also called as female gametophyte. Now how this embryo sac is formed? This is generally formed from a megaspore. So now let us come to this term. What is a megaspore? See, the process of formation of megaspores from a megaspore mother cell is called megasporogenesis. Now, first of all, this should be very much clear to you that genesis means formation. Megaspore formation, the process of formation of megaspores is called as megasporogenesis. Now, these megaspores are formed from MMC, which is megaspore mother cell. Now just see this diagram first. Now this is nucleus, a collection of cells which was there, remember, in the ovule. This is a nucleus, and here it is mentioned this is micropylar end, opening. Okay, and in the nucleus only one very large cell is present which is called as MMC or megaspore mother cell. Can you see? It has a very prominent nucleus, dense cytoplasm, right? So what is going to happen? This megaspore mother cell, it is going to undergo meiosis. And firstly, it will form a dyad, means two cells, and later on 4 cells. That is what happens in meiosis. There are 2 stages. In the first stage 2 cells are formed, and in the second stage 4 cells are formed. So because 4 are formed, you can call it as megaspore tetrad. Alright. Now out of these 4, only one of the megaspore is functional. Okay, and the functional megaspore is going to form embryo sac. So see, a single megaspore is forming the embryo sac. Such type of development, like from a single megaspore, embryo sac is formed, that is called as monosporic development. Okay, it is clear to you? So just to check your concept over here, in the last lesson, chapter 1, we had discussed about the ploidy of different cells. So can you tell me the ploidy of nucellus? It will be diploid. Ploidy of megaspore mother cell, again diploid, because it will undergo meiosis. Okay, that is why it will be diploid. Megaspore, because after meiosis the reductional division is happening and the chromosome number is going to become half, so the ploidy will be N. What about embryo sac or female gametophyte which arises from megaspore? It is also N, because from this megaspore only female gametophyte will arise by mitosis, so the ploidy will be N. This is very important. Now let us see how the embryo sac is being formed. See, the nucleus of functional megaspore, it will divide mitotically, okay, and it will form two nuclei, fine, and these two nuclei will move at opposite poles, see, like this. So this is called as two nucleate embryo sac. After this, two more mitosis will happen. So two more means one, after one this will be the result, and after the second this will be the result. Clear? See what is happening in the first stage, which is two nucleate stage. Two cells are formed and they go at the opposite ends because there is one micropylar end and one chelazal end. In the next stage again mitosis is happening, so it will form two cells and it will form two cells. Again mitosis will happen, so what will be the end result? How many cells? 8. Very good. So it will be an 8 nucleate stage of embryo sac. Now these mitotic divisions, these are free nuclear. What do you mean by free nuclear? That nuclear divisions are not immediately followed by cell wall formation. You can see over here mitosis is happening again and again, but cell wall is not being formed. So it is free nuclear, means the cells, like nucleus is dividing, but cytoplasm is also dividing, but cell wall is not being formed. So this is the end stage, which is the 8 nucleate stage of embryo sac. Now let us discuss about these 8 nuclei which are formed. Now when this 8 nucleate stage is attained, now the cell walls will be formed in the female gametophyte or embryo sac. Listen very carefully. Only 6 nuclei out of these 8 will be surrounded by cell wall. What will be happening to the remaining 2 nuclei? They will form polar nuclei. Polar nuclei means they are situated in the center of the ovule, center of the embryo sac, and that center is called as a central cell in which these 2 polar nuclei are there. Now there are 3 cells which are grouped together at micropylar end, and these collectively are called as egg apparatus. Now the 3 cells, what are those 3 cells which constitute egg apparatus? 2 of them are called as synergids and 1 is called as egg cell. And these synergids, what is the importance of these synergids? They are having certain filiform or ribbon-like cellular thickenings which will guide the pollen tubes in the synergids, and if the pollen tube is guided properly, then the male gamete is going to fuse properly with the female gamete. Now just to clarify this concept, let us look at this diagram first. So what we are talking about, this is the central cell, okay? The 2 nuclei are in the center and these 2 nuclei are referred to as polar nuclei. At the micropylar end, what they were saying, there is an egg apparatus. Yes, in the previous slide you have seen. So egg apparatus is containing 2 synergids and 1 egg cell. Clear? Then these 2 polar nuclei are there, no cell wall is there inside them, only they are floating in the center. At the base, which is this is the chalazal end, there are 3 cells again which are called as antipodal. So 3 cells at chalazal end are called antipodal. Why antipodal? Means they are opposite to the direction of micropylar end. Okay. Now see very carefully. This diagram is clear. Now just look at this diagram. What is the difference between the two? It has been turned upside down. See over here, this was the micropylar end and this is the chalazal end. Here, this is the micropylar end and this is the chalazal end. So this portion has come over here. See it properly. Synergids are there. They are having filiform apparatus, ribbon-like, or you can say finger-like rather structures, not ribbon-like. They're looking like finger-like structures, and they will guide the pollen tube towards the synergids. Because egg is there, the male gamete has to fuse with this egg. So this is the embryo sac of a typical angiosperm, which is 8 nucleated but 7 celled. How 7 celled? See, 8 nuclei are there, that is clear. See, 3 cells over here, 1, 2, 3. 4th cell is this polar nuclei or central cell. 5th is egg cell. 6th is synergid and 7th is another synergid. So there are 7 cells but nuclei are 8. Clear? Now let us come to the concept of pollination, the end part of today's lecture. Pollination. It is a process in which pollen grains from anther are transferred to the stigma of a pistil in flowering plant. So basically it is the gamete transfer. The male gamete from the stamen is being transferred to the stigma. So depending on the source of pollen, there are 3 different kinds of pollination: autogamy, geitonogamy and xenogamy. Auto means self. So self means that here the pollination is occurring within the same flower. Okay. So in the flower means both the male and female reproductive parts are there. So for self-pollination, what should be there? There should be synchrony in pollen release and stigma receptivity. Means the stigma should always be ready for taking the pollen, like in chasmogamous flowers. Okay. Another types of flowers are clistogamous. Clistogamous flowers are, they do not open at all. Okay. So if they are not opening means anther and stigma, they are lying close to each other. That is why only self-pollination can happen. No cross-pollination will happen. Okay. Examples are sunflower and pea. So some plants, they produce both types of flowers, chasmogamous and clistogamous. So you should learn the examples: oxalis, commelina, viola, which is common pansy. And these clistogamous flowers, they are autogamous. Autogamous means no chance of cross-pollination. Why we are saying this? Because clistogamous flowers are such flowers which will never open up. Means they will be closed. So if they are closed, what will be there? The male gamete of that same plant is going to fuse with the female gamete of the same plant. Okay, the male gamete or female gamete of different plants cannot fuse. That is why no chance of cross pollination. And they will produce assured seed set. See, in case pollinators are not there. What are pollinators? The organisms which can cause pollination, like honeybees are there. Okay, so if those are not there, the seeds will be formed because they can do self-pollination. Disadvantage would be that because only self-pollination can happen, so genetically superior plants cannot be formed. Okay. Next, coming to geitonogamy. In this type, pollination occurs between flowers of the same plant. Okay. Now, same plant, then how is it different from autogamy? Actually, same plant means plant is same but flower can be different. See, this is a plant. Here one flower is there. Here another flower is there. Autogamy means pollination is happening inside this flower only. Geitonogamy would mean that pollination can occur between these 2 flowers. Alright. So functionally it is cross-pollination, like cross-pollination. But genetically it is like autogamy. Because it is in the same plant, no genetic difference will be there, like in maize. Next is xenogamy. In xenogamy, actually it is cross pollination, mean pollination occurs between two flowers of different plants. So here genetic difference will be there because two different plants are there, like in onion, broccoli and spinach. So just to clarify this, just see the three types of pollination just summarized over here. See, this is a plant A. Okay. Autogamy means, look at this flower. The pollination is happening in this flower only. Okay, because male and female gamete both are present. So in this flower, the pollination is happening. Geitonogamy means we are talking about a plant only. In a plant, many flowers are there. So pollination is happening in the same plant, but different flower of the same plant. Means you can say pollen grains can come from here and attach to the stigma of this. Okay. Now xenogamy would be, this is plant B, pollination between A and B is xenogamy. Okay, coming again, autogamy means in the same flower. Geitonogamy means different flower of same plant, and xenogamy means flowers of different plant. Now there can be different agents of pollination which we will discuss in the next lecture. So just intro I am giving you here, different agents of pollination. Like there can be two types, abiotic pollinators and biotic pollinators. Abiotic means non-living, biotic means living. So wind and water are abiotic pollinators and biotic pollinators are animals. Majority of the plants, they use biotic pollinators only, and thousands of pollens are released to increase the chance event of this pollination. All right. So in the next lecture, stay tuned. We will be discussing the different agents of pollination in detail.